Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject Recombinant DNA Technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today's topic is restriction enzymes, classification and nomenclature. In this session, you will be learning about what is a restriction enzyme, the nomenclature of restriction enzymes, the major types of restriction enzymes and their characteristics, and in particular, you will be studying about type 2 restriction enzymes in detail. Let's learn about the classification and nomenclature of restriction enzymes. So, you know that restriction enzymes are endonucleases which produce internal cuts in the DNA and also at specific sites called as recognition sites or restriction sites. So, these enzymes are present in bacteria and in the bacteria these enzymes they act as immune system and gives defense mechanism or protection against viral attack. So, this we have learned in the previous session. So, coming to the nomenclature, the restriction enzymes were named based on the bacterial genus, the species and the strain from which they are isolated. Suppose EcoR1. EcoR1 is the name of the restriction enzyme which is isolated from the bacteria Escherichia coli. So, the name is represented, the restriction enzyme is named, the first letter is represents the genus name Escherichia, and the next two letters they represent the species name coli, and the next letter it represents the strain, that is the E. coli, and the strain is R, and the last one it is represented in Roman numeral 1. So, the order in which it is discovered, suppose in the Eco, that is Escherichia coli R strain, 5 enzymes are discovered, EcoR1, EcoR2, EcoR3, like that. This is the order of discovery. Okay, the nomenclature, the first 3 letters are written in the form of italics, and first letter represents genus Escherichia, the next 2 letters they represent the species coli, the fourth letter it represents the strain Escherichia coli R strain, and the last one is given in the form of Roman numeral and it gives the order in which it is discovered. Okay, these are the examples of various restriction enzymes. PVU1 means Proteus vulgaris. It is recognizing a 6 base pair sequence and the cutting is between T and C. So, it is a staggered cut and is producing 3' overhangs. Then EcoR1. So, the one which we are frequently, the example which I am frequently using. So, it is also recognizing GATT-TC sequence. It is a 6 base cutter and it is giving 5' ends. Okay. The BAM-H1. BAM-H1 means Bacillus amylo liquefaciens. It is also a 6 base cutter and 5' overhangs. It is giving 5' overhangs. Then TAC1. Thermus aquaticus, it is a 4 base cutter, it is giving 5' overhangs. Next, HAE3 Haemophilus aegyptus, it is a 4 base cutter and is having blunt cuts, it is producing blunt cuts. Next, the Haemophilus parainfluenza, it is also a 6 base cutter, it is also producing blunt cuts. And the last one, Nocardia, is a not one, it is the only, that is we have said that the restriction enzymes are most commonly seen in bacteria, and rarely they are seen in certain fungi, like this nocardia is a fungus. The restriction enzyme is NOT1. It is able to recognize this 8 base pair sequence. It's a 8 base cutter and is producing sticky ends with 5' overhangs. Coming to the classification, it's very important. The three major classes are present in restriction enzyme. They are classified on the basis of the type of sequences they are recognizing and the nature of cut they are making. These and also on the enzyme structure. Based on the enzyme structure also, the restrictions enzymes are classified into three types: type 1, type 2 and type 3. So, of all the three types, the type 2 enzyme is the one which is most prominently used in gene cloning experiments. So, let us first learn about the type 2. So, these enzymes, that is we have know that the restriction enzymes, it exists in combination with the modification. Methylase function is also seen. So, here they exist as a separate enzymes. Restriction cleavage property is present in one enzyme and methylase property is present in another enzyme. They exist in separate enzymes. So, it is possible to cleave the DNA even in the absence of modification. And these type 2 enzymes, they recognize generally 4 to 8 base pair. The recognition sites are either 4 base pair, 6 base pair or 8 base pair. So, 4 to 8 base pair sequences are recognized by type 2 enzymes, and whichever site they are recognizing, they produce cut within that site. Okay, they are highly site specific and in nature, and they cut within that specific site. And the sequences are also, the recognition site sequences, they are also palindromic in nature. Palindromic means if you read the sequence from left to right or from right to left or from 5' end to 3' end or 3' to 5' end, they read the same. For example, Madam. So, if you read from left to right or right to left, it reads the same. Malayalam, Liril. So, these words, they are called as palindromic words. So, the sequences present in the restriction sites, they are palindromic in nature. So, for this cleavage function, they require the cofactors like magnesium, ATP. Just this only cofactor, that is magnesium itself, is enough. No more energy, that is ATP, is not required for this cleavage. So, it is highly, that is economic in function. Then these type 2 endonucleases, they are most commonly used in gene cloning experiments because of its high specificity and high specificity in cleavage functions. Okay. Coming to the type 1. These type 1 enzymes, they are highly complex. They are having 3 subunits. And each subunit will be, so one subunit will be recognizing, and one it will be cutting the site, and another unit it will be showing the methylation function. So, because all the subunits are present in the same enzyme, they are highly complex, and also they recognize certain sites and cleave away from the recognition site, 1000 base pairs away from the recognition site. So, they are non-specific in cleavage, non-specific in cutting the recognition site. Also the recognition site is asymmetrical. They are not having symmetry. Next, they are also high energy consuming, that is they require, apart from this magnesium ions, they require ATP and adenosyl methionine, they are required. So, they are also highly energy consuming enzymes. So, they are not preferred in gene cloning experiments. Coming to the type 3 restriction enzymes, they have 2 different subunits. that is the recognition and modification is done by one subunit, whereas cleavage is done by another subunit. They are non-specific in nature, that is they recognize certain sites but produce cuts at another site, that is 24 to 26 base pair away from the restriction sites. So for this reason these are also not preferred in gene cloning experiments, and they also require high energy for cleavage. So you will be seeing the overall view in this table. That is the first type of restriction enzyme, ECO K1. Example is ECO K1. They are asymmetric and complex. They are non-specific in cleavage, that is they cut 1000 base pairs away from the restriction site. They exist as a single enzyme, and 3 separate units are present. So it's called as heterotrimer, and it requires cofactors and energy. For this reason it is not preferred. And the second one, let's see the third one. Third restriction enzyme is, example is ECO P1. The recognition sites are short. They are also asymmetric and non-palindromic. The cleavage site is also non-specific, that is they will be cleaving 25 to 28 base pair away from the restriction site, and they exist as heterodimers, that is 2 subunits are present in this enzyme, and it is also requiring energy for cleavage. So for this reason they are not preferred. Coming to the second restriction enzyme, this example is ECO R1. They are short, the sequences are short, the recognition site is short, that is 4 to 8 base pair. They are having symmetry. Symmetry means whenever you are cutting exactly in the middle, you will be 4 base pair sequence means 2 2. 6 base pair means 3 base pairs you will be getting. 8 means 4. So they are symmetric, palindromic in nature, and they are highly specific, that is they recognize certain sites and produce cuts within those sites. So this function is the it is important, and they exist as separate enzymes, that is the methylase and cleavage nuclease function is existing in a separate way. So it's called as homodimer. Then it don't require any energy, just with the support of this magnesium ions cofactors, the reaction will be taking place. For these reasons, for this all favorable reasons, this type 2 restriction enzymes are extensively used in gene cloning experiments. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.